Okay. Hello, everybody, and we're really happy to be showing you some of our data. I really wish we could have done it in person, but I think it's really safe to do it that way, and I really hope all of you are taking care of yourselves and being safe. Today, I'm going to show you some of the data that is coming from our Baylor groups, which is the groups of Peggy Goodell's and Rachel Rao, which will join me in a minute. And we'll talk about some of the things we're doing about TBRS here in Baylor. So before I get started, let me introduce you to our group. This is, so this is our group. It's a group of Peggy Goodell right here in the middle. This is me, and this is Dr. Rachel Rao. She's a pediatric hematologist, and I'll introduce her later as we go along. So we are all located here in Houston, Texas at the Baylor College of Medicine, and Rachel is also part of the Texas Children's Hospital. And the things we are working on is with DNMT3A are really many of the aspects of DNMT3A, and this has been occurring in Peggy's lab for maybe the last decade or so. And you probably heard a lot from Kate and Marwan and others about the roles of DNMT3A in TBRS, but I think I want to start by explaining a little bit why is DNMT3A such a big and important player in our cells. And before we talk about cells, let's talk a little bit about what are cells and how everything is affected by DNMT3A. So if this is an organism or us as people, we have tissues, and we have different tissues in our body. Here you can see our bone marrow, a liver, and a lung. But all of those tissues hierarchies or our architecture are really compiled of cells or different cells. And in all of those cells, we have proteins, and the template or the instructions to make those proteins that make everyday function for our cells in everything you can think of is coming from DNA. And the reason DNMT3A is really important is because DNMT3A changes the way our DNA is read or the way our cells interpret our DNA in each of their cells. And we know a lot about DNMT3A from years of research, and we know it's really important for embryonic development, for the silencing of chromosome X, but what we also know is that DNMT3A is really crucial and for stem cells. And stem cells are really special cells because those cells are really naive or really primitive cells that give instructions to make more specialized or more mature cells. And they are really present in various tissues in our body, as illustrated here. And when we lose DNMT3A, what happens is you can imagine losing the master regulator who tells the DNA that changes our DNA, and then vice versa, changes our proteins and our cells and our tissues. So when you think about it, it's really like losing a control tower in an airport, a busy airport, and now all the planes don't go nowhere to go. And that means that for our cells, you change the way the stem cells make other cells in the tissue. And that's why you also change the way the tissue responds. And when I say tissue respond, you can think about many things. We sleep, we eat, we need to respond to inflammations, we need to respond to viral infections. Our cells are always active and are always responsive. And if you change that a little bit, it might change the way the cells respond. And we'll talk about that later on. So before I really show you the data, I want all of us to be on the same page when we talk about what is affected by DNMT3A and what we know about DNMT3A. So probably I would imagine that Kate showed some of this already and discussed some of this with you earlier today. But I want to talk about the different mutations we have in DNMT3A and how they affect all of us, not just TBRS, all of us humans. And some of the mutations that are affected with DNMT3A can be somatic mutations, which are mutations that we can get later in life and are not passed to our children, versus germline mutations, as in the case with TBRS, that occur very early in life in the embryo stage and can be passed to our offspring or our children. So the diseases that are associated with DNMT3A and that have been studied very broadly are the occurrence of DNMT3A mutation in aging. And this is normal human aging. And I think Rachel will talk about that a little bit later. And so those are somatic mutations that we accumulate in our blood and they increase our chances to develop cardiovascular disease and anemia, and you'll hear about it more. But what we also know about DNMT3A is that somatic mutations in DNMT3A are really strong and really high in the blood system, where they can also lead to malignant transformation and blood cancers. So the case of TBRS is really different because in TBRS, the mutation is a mutation that is germline, which means that it's in all of our cells, it's in all the cells and can be passed to the children. And the symptoms of TBRS are really different from the other somatic associations with DNMT3A, which is mainly the blood system, as where it affects overgrowth, obesity, and developmental delay. So our goal here in Baylor is really to understand how DNMT3A mutations impact all the life of TBRS. And it's really a slow process, and we're only in the beginning. And that's why I have a letter here, because it's really years of research ahead of us. And we're only just now beginning to unfold and unravel some of the things that are affected by this germline loss of DNMT3A, which is different from what we know of the blood. And another thing which is really important for us here is to identify intervention strategies or use the research we do here with your help to try and improve the life of TBRS individuals. So how do we study TBRS? And we really want to thank all of you who are part of this study of TBRS and who help us with this study of TBRS, which all starts with you giving us a little bit of your children's or yourselves blood. And with this blood sample, what we do, we can isolate different populations. We can look at different blood populations. You will hear about that a little bit later also. And we can also make cell lines. And what those cell lines are cells that are extracted from the blood that we can grow, as you can see here in a lab setting, in a plate. But what we also do is we make mouse models that recapitulate that they are the same mutation we see in the human to the mouse, which tells us a lot about the behaviors. And we know that mice and humans are not the same, but mice, because they're really in a really laboratory setting, really help us to see some of the phenotypes and study them further in a really controlled environment. So that's why mouse models are really important. And with your really gracious help, we were able to publish two publications, major publications in the year of 2020 or 2021. One of them is the study of mosaics, and we'll talk about that soon. And the other one is the impact of the changes in blood in TBRS individuals that Dr. Rao will tell you later. So our research questions that are the topics I'm going to cover today are how is blood production changing? As we talked about, as I told you in the beginning, we know a lot about somatic mutations and how somatic mutations in the blood impact blood development in aging and in cancer, but we don't really know what it means when you're born with the DNMT3A mutation very early and how does affect blood production. I want to tell you a little bit about DNMT3A mosaicism. Why is that important? Why is that those individuals that are mosaic can really help us learn so much about or other individuals with TBRS. I'm going to talk about a little bit about the weight regulation, which I know is a major concern from some of the parents here. We'll talk about what we're doing and what we think should be done next. And finally, I want to tell you a little bit of another study we're doing where we try to understand how each of the mutations is different. And although we see many symptoms or many associations with TBRS between children or between individuals that are really quite similar, but the mutations are really quite different. So we want to, so I'll show you how do we interpret this and how do we use the different mutations to understand something about DNMT3A. So let's get started. And the first topic I would like to talk to you about today is the DNMT3A mosaicism. So what is DNMT3A mosaicism and what is mosaicism? So we talked before about somatic mutations that occurs later in life and the germline mutations that you're born with and can be passed to your children. A mosaic mutation is a little bit different. It's a mutation that still occurs very early, but it's not evenly spread within in all the cells in your body, which means that some of your cells are really retaining a normal copy of DNMT3A, while the other cells here, for example, here in red, are cells that have a mutated DNMT3A. And of course, these ratios can change, and some tissues can have more, some tissues can have less, and I'll show you what it means and what we think it means. So the reason we think mosaics are really important is because one of the studies where we studied, we came across an individual that was mosaic. And the reason we found this individual because he had 14 kids and 4 of them had TBRS, but the other kids had a normal copy of DNMT3A. And what we saw about this individual, he was 68 at the time we started the research on him, and he was really healthy. A 60-year-old man with a mixture of cells in his body that are wild type or DNMT3A mutated as in TBRS. And what was interesting about this individual is that it was the first time we could actually see how different tissues in the body or different tissues where we were able to access have different ratios of DNMT3A mutant versus DNMT3A wild type. And what we found was really quite important when we looked at the blood, and this is a ratio of the mutation in those tissues or cells. And because we have in our cells 1 copy of a mutated DNMT3A and one normal, 50% or nearly 50% means that almost 100% of the cells are coming from a DNMT3A mutant source. And it's really quite striking, but you can see it here, that if you look at DNA from eyebrows or saliva or urine, they're really quite low compared to the blood, where we can see in this individual that I really want to emphasize is really quite healthy. It's almost 90% of his blood coming from a DNMT3A mutant source. So what does it mean? Why is DNMT3A mutation taking over the blood? And what does it do? It's still something that we really want to pursue further. And is this unique to this type of mutation, or is this going to be different with different mutations? Will different mutations in DNMT3A in mosaic individuals have different capabilities to take over different tissues? It's something that we still don't know. But we think that by learning from those individuals, and we really urge those individuals to come forward and volunteer to participate in the study, we can learn how DNMT3A mutations change our tissues differently. So this is one of the things that we're still working on and trying to understand. But because we had this model of TBRS, one of the things we really noticed is when we started to make mice with TBRS, as for some of the individuals, we saw that some of the concerns are really weight gain. And in the mice, it was really quite striking. And this is a control mouse that has a normal DNMT3A. And this is a TBRS mouse model. And you can see that by eye, that the weight of the mice is really quite big. The mice develop a lot of fat tissue. This is a CT scan of the mouse. And you can see all of this in gray is the fat tissue and abdominal fat. And you can see that there is a lot of fat being developed in those TBRS mouse, and this is on the expense of our lean tissue. So something in TBRS is changing the shift between fat production and lean mass production, which is like other cells like muscle and bone. So how do you gain weight, and how is weight regulated? And I'm going to keep it really, really simple, but it all starts with us eating or consuming food. So when we eat, all this fat, for let's say we eat a really cheesy, nice pizza, all this fat from our food has to be stored somewhere so it doesn't disturb other normal tissue functions, just as the heart. So the way our body deals with it is that we have our adipose tissue, which is our fat tissue, where all of those lipids or fats are stored in lipid droplets. And when we have enough, we ate enough and we have enough storage of fat in our body, what it happens is that our fat cells go and release molecules that tell the brain, hey, we have enough fat, please stop eating. And then what the brain releases a different molecule that talks to the fat and says, okay, everything's fine and this is well balanced. And in oppositely, when you don't have enough fat, let's say we're after a sleeping cycle, like every time we wake up in the morning and we're hungry, it's because the brain knows that there is a decrease in our fat, and there you have a cycle of communication between the brain who controls our feeding to the fat tissue that stores our fat. So in the mice, this process is really easy to monitor and control, and it's really easy to monitor how the brain responds by controlling the feeding and how the fat tissue responds by storing the fat. So we took our TBRS mouse models here in red, and we basically just waited over time and measured how much they eat over time, over the course of a few days. And what we saw constantly is that our TBRS mice just consumed a little bit, not a lot, but a little bit more food than our control mice. So meaning that our TBRS mice eat more. So if they eat more, what the brain should now do is tell our fat, enough, we have enough. So, but that means that they eat more. It means that something in this control between eating and storage is now working properly. So what about the storage? To check the storage of fat, we took cells that are fat cells and we removed DNMT3A here in red. And what we noticed, first of all, is that when you remove DNMT3A from fat cells, you get more fat cells. They grow more. So as you can see here, you look at the red compared to the black, you see that the red is higher, which means that they make more cells. But in those cells, which they make more of them, what happens to the fat? Because as we said, they eat more, so they should have more fat. But what happens is that we look at the fat inside those cells, and now we look again at the black compared to the red, we see that the black has more fat cells. Our controls, our normal cells have more storage. That tells us that inside the TBRS cells, although they have more cells, they don't use it efficiently and they don't store as much fat. So in TBRS, although they have more cells, they have less fat in those cells. So if I summarize this part, in a normal mouse, what we see is that there is a communication between the brain and in the fat tissue where we control the number of cells or the number of fat we need to store, and the brain controls the feeding to regulate how much we need over time. In TBRS, there is no communication. The brain doesn't know that there is enough fat, and the fat doesn't release to the brain signals that tell it no more feeding. So something is broken there in between the communication between the brain and the fat tissue, where the brain knows that there isn't enough fat, so and the fat cells are less efficient. And if you go back to what I told you in the beginning, where we know about DNMT3A and how DNMT3A changes our DNA and our protein in our cells, this is a really good example, because although we have a really good tissue, we have fat and everything is working, the cells are not perfect. The cells are not as responsive as a normal cell or a normal tissue. The next topic I want to talk to you about is how do we separate the different mutations? How do we learn about each of those mutations in TBRS, which is really remarkable that when you think about all DNMT3A, and DNMT3A is a really big protein or a big gene, and it has a lot of domains, and each of those domains does something else in the cell regarding to the DNMT3A function. And as I told you, this affects, of course, how the DNA is read, how the proteins are made, and how the cell and the tissue respond. So in TBRS, we have mutations all over those three domains, and each of them are really different. But what does it do and what does it mean? And something that we know from many years in biology is that the cells are really smart, and if a protein they feel is bad, or something in the protein doesn't seem right, the cell have a mechanism in which they dispose of that protein. So in each of us, we have two copies of DNMT3A and, that we get, one from our mother and one from our father. And in TBRS, usually the case is that you still retain one normal copy and then you have another mutant copy. So we have two. But in those cases where the cells thinks that the protein, the DNMT3A mutant protein is bad, what will happen is that it will degrade the mutant copies. So instead of having two copies of DNMT3A, we're left with one. And it's very similar like having the whole DNMT3A removed, because now we only have half the dose of what we are supposed to have. So this is one of the projects that a very talented now postdoc in Stanford, Shin from our lab, did, and he really wanted to understand what happens to the proteins of DNMT3A in each of the mutations. So he not only just explored TBRS, he took all the mutations we know about DNMT3A from also somatic mutations as occur in blood aging and blood cancers. And he made a library of all those mutations, and he asked a really simple question. Will the cells, when I introduce this mutation, will the cell think it's a bad mutation and degrade it, or will this mutation make the DNMT3A stay in the cell, so you have two copies? So if this is our arbitrary line of unstable, we call it, that's a mutation that will make the protein go away. And mutations that are above this red line are mutations that will retain the DNMT3A. It was really quite striking that many of the mutations in TBRS and others are mutations that basically lead to the fact that you have only one copy of the DNMT3A, so half the dose. So this is really giving us an avenue for therapeutics, because if we realize, or if we understand how to make the copies be stabilized, we can maybe control the protein level of the DNMT3A and improve the outcome on patients. So if I summarize what I've showed you today, I've shown you that it's really important to have each of you investigate, and each of you should, if you really want, contribute to our research, because we can really learn from each of the mutations, and each person with TBRS is really different. I've shown you that in TBRS, there is a miscommunication between the brain and the fat tissue. That leads to weight gain and overeating. And I showed you that by studying a mosaic individual, we found that DNMT3A mutations take over the blood system. So it's really important in the blood, but why and how is something that we still don't know. And then I've also showed you that about a third of the DNMT3A mutations we investigated affect the protein in a way that the DNMT3A mutant protein is degraded or disappears from the cells, leaving the cells with only one copy. So I know you probably have many questions, and I'll see you in person in the live chat probably after this talk. So I'm really looking forward to it, and I hope you're enjoying your time here at the conference. So before I finish this, I would like to introduce Dr. Rachel Rao. She's a pediatric hematologist oncologist, and she's going to talk to you about our research of DNMT3A in the blood, and maybe illustrate for you how blood development is affected by TBRS. Thank you.
Hello, everyone. Thank you so much, Ayala, for a fabulous talk and a really nice introduction. And I'll start by sharing Ayala's wish that we could all be together in person, but of course, safety first. And I hope everyone is safe and healthy, and we'll hope to see everyone this time next year in person. So as Ayala said, I'm a pediatric hematologist oncologist. And essentially what that means is I'm a physician who cares for children with disorders of the blood and or cancer. And that's my clinical position is at Texas Children's Hospital. And then I also do research in the lab focused on these disorders of the blood and cancers in children. So why we were even beginning to be interested in DNMT3A is because of its role in the development of blood. And so first I'm going to have to start by explaining what blood is and how it develops in the first place. So the blood is actually a very remarkable system, and the development of blood occurs through this very well-regulated stepwise process in which all the normal cells of the blood are formed. And this process starts with this very specialized cell that we refer to as a blood stem cell. A blood stem cell resides in our bone marrow, which is the factory for production of all blood cells. And it has the remarkable capacity to not only make new copies of itself, but also to differentiate into and form all of the mature blood cells that we need to survive. And this process goes down a path by which the cells gradually mature into sort of these earlier starting to become a mature cell, all the way down here to where we formed all the cells that go from the bone marrow out into our peripheral blood to perform very specific functions. So sort of walking through what all of these functions are, we'll go one by one through these different cell types, starting with what we call erythrocytes, or more commonly known as red blood cells. These are the cells that are responsible for picking up oxygen in our lungs and carrying it and distributing it to all of the tissues throughout our body. So obviously a very important function that is necessary for the functioning of every part of our body. The next little cell here are called platelets, and blood platelets are actually fragments of cells that are responsible for controlling bleeding. So if you get a nick or a cut, the platelets are the first thing to go to that cut in the blood vessel and seal it up to prevent catastrophic bleeds. So obviously a very important blood cell for maintenance of life. And then this last group here is made up of a lot of different types of cells, collectively referred to as our white blood cells. And white blood cells are the infection fighting part of our blood. These are the cells that recognize a pathogen, be it a virus or a bacteria, and help to get it under control and clear it from our bodies. So that's normal blood development. Now, obviously, this process is usually well regulated and well coordinated, but things can go wrong along the way that lead to abnormalities of our blood. And there are a variety of different abnormalities that can occur in our blood. If something goes wrong with the red blood cells, for instance, perhaps your body isn't making enough of them, that condition is referred to as anemia. And the problem with anemia is you don't have enough red blood cells picking up that oxygen and carrying it to all the cells in your body. Obviously, if you don't have enough blood platelets, you could be at risk for having a significant bleed. Conversely, the blood platelets can be abnormal in a way that makes you more likely to have blood clots. So multiple different issues can arise with the blood platelets as well. And then lastly, of course, because the white blood cells are responsible for fighting off infections, if we don't have enough of them, or if they aren't working properly, then we can be at risk for development of severe infections. Additionally, these are the cells that sort of help clean up any damage to any tissue. And that process can lead to inflammation. And it's possible, inflammation to some degree can be helpful to the body, but when there's too much of it around, it can lead to excessive damage to specific tissues. Additionally, white blood cells can cause a very annoying allergy. So the seasonal allergies that we get when the pollen is high is mediated by these white blood cells. Additionally, when the white blood cells are too active, they might start to recognize parts of our body as being a foreign invader. And this process is called autoimmune issues. And you might know individuals with things like lupus or rheumatoid arthritis. Those are because our own immune system is recognizing parts of our own body as foreign and trying to get rid of them. Now, of course, perhaps the most extreme example of something that can go wrong with the blood is the development of a blood cancer, more commonly referred to as leukemia. And there are a few different ways in which a leukemia can develop. And the types of leukemia in general broken down into two major subsets. One type of leukemia is called acute myeloid leukemia, and that's leukemia that has developed in a blood cell that was destined to become a specific type of cell called a myeloid cell. Additionally, acute lymphoid leukemia can develop if the leukemia arises from a cell that was destined to become a lymphoid cell, which is another specific type of white blood cell. And these usually occur very rarely in children, but can happen if some deleterious change, so something bad has happened to the DNA of the blood cell that has caused it to lock into the state where it is incapable of maturing down this stepwise progression to becoming a normal functioning blood cell. And the changes to those cells make them grow very rapidly. So they rapidly sort of overtake all of our normal blood production, crowding it out so that you can't make all of those normal cells downstream. So, I think a big question for a long time is blood production affected by DNMT3A mutation? And really, this is one of the things that got me even interested at all in DNMT3A because of its known sort of an emerging role in the regulation of normal blood production. And perhaps, this really emerged around 2011, 2010, so now over 10 years ago. In Peggy Goodell's group, they had identified that if you lose DNMT3A in the blood cells in mice, the overall the blood system was reasonably normal, with the exception of you just were having those blood stem cells making more and more and more copies of themselves and not really effectively differentiating into all the mature progeny of cells. So a sort of imbalance between the blood stem cells making new copies of themselves and making those mature downstream copies. Around the same time, Tim Lay's group in WashU identified mutations of DNMT3A, specifically in blood cancers, first described in the blood cancer acute myeloid leukemia, and subsequently identified in a number of other blood cancers. And you can see the relative frequency. If you take all adult patients with acute myeloid leukemia, for instance, a little over 20% of them will have a mutation in DNMT3A. Now, what's interesting is that while DNMT3 mutations are common amongst adults with specific blood cancers, and you can see those sort of represented in these bar graphs down below, DNMT3 mutations are almost never identified in childhood cancer, blood cancers in particular. And you can see the rates here are, some are almost 0%, upwards of, maybe 2% in a specific subtype of acute leukemia in children. So this suggests that while these mutations may play a role in the development of cancer, it probably takes a really long time for a blood cell that has a DNMT3 mutation to grow into a cancer. It probably also means that multiple other problems have to occur in those same cells to ultimately make it go from being a blood cell with just a DNMT3 mutation to a blood cancer. I think that sort of idea of the fact that it would take a long time for a blood cell with a DNMT3 mutation to grow into a cancer or to change into a cancer is further supported by something that Ayala had alluded to in her talk. There have been a number of studies where they take blood samples from otherwise, healthy people without any obvious blood abnormalities and do a sequencing test looking for DNMT3A and other mutations. And what they've identified is that a lot of regular, otherwise healthy people without any blood abnormalities whatsoever will have DNMT3A mutations present in some of their blood cells. In this process, we've termed clonal hematopoiesis, clone meaning it came from one cell, and then hematopoiesis referring to blood. So if you look at actually regular people, healthy people, no blood abnormalities who are of the age of 40 to 50 even, and you sequence enough cells, millions and millions and millions of cells, in the vast majority of individuals, you actually find some presence of DNMT3 mutation in the blood. Now, in a small subset of those individuals, that percentage of blood cells that have a DNMT3 mutation will gradually increase over time. And this is what you see here. So if you look at 70 to 80-year-old individuals, you'll see the presence of some of these mutations at or above 2% of the blood cells in around 10% of individuals. And that gradually increases over the lifespan. So this increases with aging, that DNMT3A mutations will gradually increase in the blood of certain individuals. But again, if everybody by the age of 50, you can find a DNMT3 mutation, but only 10% of people by the time they're 80 will have 2% or more of the blood cells having DNMT3 mutations, that means this only happens to a subset of individuals. And there's a lot of ongoing research to help identify what puts you at risk for that expansion of DNMT3A in your blood, but we still don't have a good understanding of that. So obviously the next logical question is, if you have a DNMT3 mutation in the blood, does that predispose you to the development of those blood cancers where we know DNMT3 mutations can occur? Well, the answer is it does probably increase your risk a bit because we know that individuals with this DNMT3A mutation present in their blood, a certain percentage of them will develop a blood cancer, and that's slightly higher than the general population, than individuals without a DNMT3 mutation detected in their blood at that 2% or higher threshold. But still, the vast majority of these individuals will lead otherwise healthy lives without the development of a blood cancer. Additionally, Ayala mentioned the father of the children with DNMT3A, and 90% of the cells in his blood have a DNMT3 mutation, and he is otherwise healthy. He's aged over time and hasn't developed a blood cancer. So certainly the presence of a DNMT3 mutation in the blood doesn't mean that you'll definitely get a blood abnormality, and certainly doesn't mean that you'll definitely get a blood cancer. In fact, a small minority of individuals with DNMT3 mutations in the blood go on to develop a blood cancer. Now, it doesn't mean it's not problematic at all, because we do know that individuals with DNMT3 mutations in their blood are at risk for other issues. Probably most importantly is cardiovascular disease. Coronary heart disease seems to be higher amongst these individuals. And we think that's due to the inflammation that the abnormal cells, the DNMT3 mutant cells, create. Another, I think, important piece of evidence suggesting that DNMT3A might impact blood development comes from Dr. Tatton-Brown's paper, a really nice and comprehensive overview of 55 individuals with TBRS, with really nice descriptions of the health issues that they've experienced throughout their life to date. And if you look through that really nice table in the paper, she's very clearly documented a number of things that suggest there may be abnormalities in the blood. 4 of those 55 individuals had some mention of recurrent infection. So perhaps, that army of white blood cells that we all normally have to fight off infections is somewhat less than in individuals without TBRS. Additionally, in 3 of the 55 individuals, there's a note of a bleeding or a platelet issue, suggesting that perhaps that type of blood cell is somewhat affected by the presence of a DNMT3A mutation. And then, of course, 1 out of the 55 developed an acute myeloid leukemia at the age of 12. So all these things led us to wonder, what impact does DNMT3A mutation have on blood development in individuals with TBRS? So every cell in the body having a DNMT3A mutation, how does that impact blood development in these individuals? Does it misdirect the blood? Do you develop too many of one type of cell, too few of another? Do the cells work properly? So these are the questions we wanted to start answering. Additionally, since we know those white blood cells are really important and they're immune cells fighting off infections, are there differences amongst them in individuals with TBRS? So to do this, we set about to analyze the blood of individuals with TBRS. And as Ayala said, a huge thank you to those who contributed to this research. We know it was asking a lot of you to have blood drawn from your children and their siblings and shipping them sometimes all over the world to participate in our research. And with this blood, we did a number of analyses. We really wanted to define how many of each of those different types of white blood cells were present in the blood in the TBRS individuals compared to individuals without TBRS. And the way we did this was a special test called flow cytometry, where it takes a tube of blood, just even a small amount, and then we can add a stain to it that tags blood cells, sort of defining them as red blood cell versus a specific type of white blood cell, and really allows us to tease apart how many of each individual type of cell in a person with TBRS compared to a person without TBRS has. And this work was done in collaboration with 2 of my colleagues at Texas Children's. Dr. Andrea Marklees is a pathologist, and then Dr. Amos Gawad, he actually does all the flow cytometry assays for all of Texas Children's. So they have a great deal of expertise in this analysis. So who participated? This is quite remarkable, in my opinion. We were able to collect samples and data from 18 individuals with TBRS, and the age range from 2 years old all the way up to 38 years old. We could do our analyses on the vast majority of these individuals. 15 of them, we were able to do the flow cytometry analysis. And then 13, we were also able to do blood cell counts. So basically what we call a CBC, to look for basic blood parameters. Additionally, having the medical records contributed from a number of individuals helped us to track some differences over time. Additionally, we enrolled 11 individuals without TBRS, and the age range was pretty comparable, 3 years of age to 4 years of age. 7 of them were siblings of individuals with TBRS, and then 4 were not related, just otherwise unaffected children. So this is just a map showing where all these samples came in from. We're here in Houston, Texas, as Ayala said, and we got samples really from all over the world. I think 5 different countries and several states contributed. And since the publication of this study, we've been contacted by individuals in a number of other countries as well. So it's nice to see the global network contributing to this research. So what do we find? I'm going to break this down bit by bit and review some of the highlights of the result. But I think most importantly, what we found is that by and large, individuals with TBRS have blood results that are within a normal range. I think the differences we saw, whereas the individuals without TBRS really clustered in the middle of that normal range, individuals with TBRS tended to cluster on one side or the other of the normal range. But I think it's really important to note that by and large, the results we got were within the normal range. What we found were some differences within that between individuals with TBRS and those without. So I'll review some of those results and sort of highlight what those differences were. So starting with the red blood cells, we were able to find that overall patients with TBRS were not characterized by having anemia. They had normal numbers of red blood cells. And then another sort of useful metric we look at from red blood cells is the amount of hemoglobin. And hemoglobin is the part inside the red blood cell that actually picks up the oxygen and carries it throughout our body. And those values were not different between our TBRS individuals and our non-TBRS individuals. And you'll see in red our TBRS, and in gray, each dot represents one individual, our controls, those without TBRS. So they were not anemic. Another metric that we look at to understand the red blood cells are how big are they? The relative size can vary from one individual to the next. And what we found is that the red blood cell size of individuals with TBRS was on average higher, bigger than red blood cells of individuals without TBRS. Now, let's say that this is sort of the range of normal that the test has established, down here being a smaller but normal size red blood cell, and up here being a larger but still within the normal range red blood cell. The siblings and the other individuals without TBRS tended to cluster right around the middle of that normal range. You can see that here. Whereas our individuals with TBRS sort of shifted. They were on the upper end of that normal range, with just a few sort of touching on right at the edge of that normal value. So in summary, from the red blood cells, TBRS individuals tend to have larger red blood cells, but this is not associated with anemia. Interestingly, this is something that we see in another genetic syndrome called Down syndrome, a relatively common genetic syndrome in our population. Those individuals also have larger red blood cells. In Down syndrome individuals, this is a completely benign normal finding. So we think that it's similar to what we see there. We think this is probably a benign finding, but it's important to know what your normal is. Because if you sit on the edge of that normal range, it would be important that if you get sick at a time down the road and your doctor sends off this blood test and finds that your red blood cell is slightly larger than the average, it would be important to be able for the doctor to know that that's just what's normal for you, rather than pursuing a lot of testing to understand it. So the next set of cells that we noted a few differences between our TBRS individuals and our non-TBRS individuals were in the white blood cells. And again, there are several different types of white blood cells, each performing a specific different type of function in our immune system. Most of them were not different, TBRS versus non-TBRS, but there were a few that were different. The first difference that we noted is in our TBRS individuals, we noticed an increase in the number of a type of white blood cell called neutrophils. You can see here our TBRS individuals compared to those without TBRS. So the blood neutrophil is a very important cell. It's the first line of defense against bacteria. Bacterial infections can invade our body, and when they come in, they can cause a lot of damage, and this releases inflammation from the tissue, which is sensed by the neutrophils as a danger signal, springing them into action. Neutrophils are able to catch and destroy those bacteria. The neutrophils can also release their toxic contents to break down the bacteria sort of outside of their cell, and then send off other signals to say, let's make more neutrophils and sort of get recruited to the area. So that's really important, and that's how the neutrophil was designed to fight off bacteria, but that's not the only signal that can trigger the neutrophil into action. So we know that other sources of inflammation can activate the neutrophil in the exact same way that it's designed to attack bacteria. And one big driver of inflammation that has come to light over the last several years with a lot of research is obesity. Obesity is a big source of inflammation. So an obese individual has higher sort of basal inflammation. That too could be sensed by the neutrophils, leading to this downstream cascade, which would result in the production of more neutrophils. So perhaps the obesity that we see in TBRS could be linked to this increase in the neutrophils that we saw in our lab test. Okay, and then finally, one of the notable findings from our study were in another type of white blood cell called B cells. And what we found is that our TBRS individuals, on average, they had lower numbers of B cells compared to individuals without TBRS. Now, the B cells are another really important infection-fighting cell, and their general role is to produce, is to respond to immune system stimulation, be it by an infection or a vaccination. What happens when a B cell has been stimulated is that they form the mature cell, which produces infection-fighting antibodies. And this again is in response to, say a viral infection or a vaccination that you've gotten. The net result is to produce these really important infection-fighting antibodies. Additionally, when that B cell has been stimulated, it also produces copies of itself that's capable of recognizing that same infection or vaccination if it's exposed to it again. So this is called the memory B cells. So it remembers that specific pathogen, be it a virus or other infection. So if your body's exposed to it again, these memory B cells are capable of recognizing it quickly and mounting that immune response really rapidly. So these low numbers of B cells could mean that individuals with TBRS produce less of this infection-fighting antibody upon exposure to a potential infection. This could also possibly explain some of that increased susceptibility to infection that Dr. Tatton-Brown reported and identified in her very detailed study of those 55 individuals with TBRS. Could this also mean that individuals with TBRS are less likely to mount a really vigorous response to vaccines? I think that's a super important question, one we don't fully have an answer to yet, but is worth thinking about in our individuals with TBRS. Okay, so the next part of our work was to say, well, we've identified these potential differences in individuals with TBRS versus not without TBRS. Do we see these same findings in the mice that we've created in the lab to recapitulate the findings of TBRS? And so we used 3 different mouse models. Oh, and this really nice cartoon here was generated by collecting the mutation data from individuals who've contributed to the TBRS registry. And Carrie and Jill have done a remarkable job at collating all this information and would pass it along to us so that we could really generate a pretty clear picture of all the mutations that we've identified so far in TBRS. And thanks to all of you who submitted information to contribute to this as well. And as Ayala pointed out, these mutations really can occur almost anywhere in this large DNMT3A gene. Certainly, clustering around these specific regions are what I refer to as domains. So we wanted to sort of create different mouse models that represent different parts of the gene potentially being mutated. And so we created a mouse which has a mutation in this first domain. And this is going to be referred to as our HET293, HET meaning only one copy of the DNMT3A gene has the mutation, the other is unaffected. We had another mouse with a mutation in this region 2, our 577 mouse. And then the third model we looked at has deleted one entire copy of DNMT3A. So that would represent individuals who have deletions of one copy of DNMT3A, but also, as Ayala pointed out, many of the mutations across the gene lead to a rapid destruction of the protein. So sort of this state where you have half the dose you normally do. And that would be what we recapitulate with this particular mouse model, which is referred to as the HET mouse. And by and large, the findings that I described, we did find in the mice. So in our TBRS individuals, we saw that larger red cell size, and we saw that in all 3 of the mouse models as well. The neutrophils was increased in all 3 of the mouse models as well, just as it was in our TBRS individuals. And the B cells were similarly decreased in the TBRS individuals in all 3 mouse models. So this means that the mouse models did largely resemble humans with TBRS in many respects. Of course, I think an important caveat to point out is that all of the individuals in our study were relatively young, 2 to 30 years of age. The mice we studied, we allowed to age on the, in our mouse facility for a long time, and really they were pretty old by the time we, were doing these analyses. So we don't know yet if this aging process that we see in mice is going to recapitulate what we see in human individuals, and that will take, years to decades of research to establish. So just to sort of summarize the findings and develop some guidance or recommendations regarding those findings. So again, we know that the red cells on average are larger in our TBRS individuals. This is likely benign and very similar to what we see in some other genetic disorders. So there'd be no intervention, but really important to sort of know that normally your red cells might be larger than an average individual. Okay, so additionally, we saw that TBRS individuals have more neutrophils. Could this be due to more inflammation? Since we know obesity induces inflammation, perhaps weight control could be important here. Additionally, that increased inflammation can lead to cardiovascular disease. So heart disease assessments would be really important for TBRS individuals for a number of reasons, this being one of them. And then we talked about the reduced number of B cells. We also saw some mild differences in another type of white cell called a T cell, perhaps leading to some of that increased susceptibility to infection that's been reported amongst TBRS individuals. So what can be done? So since we have concern that there may be a less robust response to vaccinations, it might be wise to have your doctor check what we call vaccine titers. And that gives us an idea of if you've had a really good response to your vaccine or not. If not, maybe you need a booster vaccine to help bring that up a bit more. Additionally, if you're a TBRS individual who has had recurrent infections, it may suggest that your infection-fighting antibodies, also called immunoglobulins, are overall low. And there's a lab test that can be done to check for that. So that's something that you could ask your doctor to send as well if you have concerns that you're at increased risk for development of infections. Okay, so probably the question I get asked the most is this: are individuals with TBRS at risk for cancer? As I said, one of the first things we learned about DNMT3A in the blood is that it can be identified, in particular in adults with blood cancer. Of course, there have been some reports in the literature of children with TBRS, a 12-year-old and a 15-year-old so far reported with AML, acute myeloid leukemia, and then one report of a 6-year-old with T-cell leukemia. We don't really have a good handle on overall the frequency of cancer amongst TBRS individuals, but certainly something we really need to nail down, I think, with research over the coming years. In addition to the blood cancers, there have been rare reports of other cancers in individuals with TBRS. We see one in the literature that has neuroblastoma, which is a type of solid cancer, a tumor, and then another with a brain tumor called a medulloblastoma. So again, establishing if this is just a one-time situation or something that recurrently happens in children with TBRS will be really important to establish in the coming years. Can we learn something about cancer incidence from studying those mouse models? I said, a lot of the blood features that we saw in our TBRS individuals, we saw in the mice. So can we learn something about cancer risk by studying the mice? Well, again, there's a caveat. We don't know that human aging is exactly the same as mouse aging, but perhaps we can get some idea about this. And in our Hematologica paper that was published this year, and then a few papers from Tim Lay's group really looked into this question. What we found is that there is an increased risk of blood cancers in the mice with the DNMT3A mutations or deletions compared to the mice from those same birth cohorts without the DNMT3A mutation or deletion. The incidence really varied between these models. There's a pretty wide range, but overall, the vast majority of mice with DNMT3A mutations or deletions did not develop a blood cancer. Additionally, when the cancer developed, it was almost never in the younger mice, almost always in really old mice. And so we think that even in the mice, it takes a long time for cancer to emerge from a blood cell that has a DNMT3A mutation. So I think the take-home point here is cancer is not an inevitable consequence of having a DNMT3 mutation. In fact, I think the vast majority of individuals with a DNMT3 mutation, be it just in their blood or in every cell in their body, will go on to develop a cancer. That said, we still need more data to understand what is the overall risk of developing cancer in our TBRS individuals. We can't really develop good screening guidance or recommendations until we have a better handle on what is the relative risk of developing a cancer in our individuals with TBRS. And that includes, of course, blood cancers, but also those other cancers. I think we really need to capture as many individuals with TBRS with and without cancer to understand what those rates are, to understand if they develop, what age are we looking at, what specific types of cancer are we talking about, so that we can develop good guidance about screening and surveillance. So what are screening recommendations? This is, I think, a really challenging and controversial topic, not only in the TBRS community, but amongst other individuals who may be at increased risk for cancer. And it's, again, really hard to make recommendations without full knowledge of what's the overall risk of developing cancer. I think in general, as we said, because TBRS individuals can have these slight differences in their blood compared to a non-TBRS individual, it's really important to know your normal. So the best way to do that, and the easiest way to do that, is to have just a regular sort of blood checkup done by your doctor at a time when you're perfectly healthy, like nothing's wrong. You don't have the sniffles, you don't have a cough, you don't have a cold, just to see sort of what your baseline labs look like. So that then if you have symptoms that are concerning, you'll have something to compare that back to. I think otherwise, doing routine blood tests periodically, it's hard to justify that because you don't have a specific thing you're looking for. And blood tests in general for blood cancers are not great ways to screen for blood cancer development. But I do think there's value in knowing what your normal blood counts look like so that you have something to compare to should you have concerns raised in the future. Certainly, though, one of the most important things I can sort of convey to you is that it's knowing when to go see your doctor, knowing when something's different that may suggest the possibility of leukemia. And again, leukemia is going to be, I think, really uncommon, but I think important to recognize when you would need to be checked for that. So the signs and symptoms of leukemia develop because, as I said earlier, all blood cells are made in the bone marrow. And that includes normal blood cells, but also leukemia cells. So when the leukemia cells start to grow there, they grow so fast that they just crowd out all the other normal cells, leading to a reduction in the number of normal cells you're able to produce. And that's what causes the symptoms of leukemia. So because you can't make as many red blood cells as you need, you can become very anemic. And the signs that point to anemia are if you have really pale skin. And I mean like different than normal. You can sort of compare back. And I say this is a good grandma test. Grandma hasn't seen you in a while. She's like, wow, you're pale, because this is something that can happen really gradually over time that even a parent can't necessarily notice because it's such a gradual change. But someone who hasn't seen, you know, the child in a while is usually the one that says, wow, you look really pale. Additionally, people who are really anemic have really severe fatigue, just like struggle getting out of bed, can't do normal activities, are too tired to eat, kind of fatigue. Anemia also causes headaches, headaches sort of throughout the day that will go away for a while, but then just always sort of recurrently come back. And additionally, you can get really short of breath because you're not carrying oxygen around to all the different tissues of your body. You get really winded. So just like walking up a flight of stairs could be, you know, under usual situations, very easy to do. But if a person who's really anemic goes up a flight of stairs, they can get really short of breath and just have to stop halfway, sort of, you know, not and catch their breath before they can go on. If you don't make enough blood platelets, that part of the blood that helps us stop bleeding, then you can have issues with bleeding. Gum bleeding when you brush your teeth, really bad nosebleeds, and everybody gets nosebleeds every now and again, but these are like the ones that just really take a long time to stop and are pretty copious. And then easy bruising. So kids bruise, right? Every child you look at has bruises on their shins, some on their elbows. I don't know, I feel like my son has bruises all over his body, but these are often bruises in places you can't explain, like on the belly or the back. And you're like, I'm pretty sure they didn't bang themselves, you know, significantly. These bruises are also quite different if they're because you have low platelets, sometimes to the point where you can actually feel them. They're sort of raised up on the skin, and they're just usually really dark purplish and sort of expand over time. So these are usually really significant bruises, not just the ones that you get sort of on the shins because you're running around or active. And then, of course, if you don't have those infection-fighting white blood cells, you're at risk for infection. So this is when you have persistent fevers. Other signs of a serious infection would suggest that you don't have enough white blood cells being produced. Additionally, because all of the leukemic cells are in the bone marrow, that's really a confined space, it can lead to a buildup of pressure inside the bone, which leads to bone pain. And that can be to the point where you have a child who is now refusing to walk because their bones hurt so bad. And it's usually really bad leg pain, Back pain is another sort of red flag. If you have a child with bad leg pain, back pain, that would be a reason to be checked out by your doctor. Another sign that's a little less common is if you get full really easily, you take a few bites of food and you're like, I just can't eat anymore. And that's because the body's spleen is right by the stomach, and it gets large with leukemia and compress the stomach, making it sort of a smaller space for the food to get into. And then lastly, rare blood cancers can lead to a tumor in the chest, and that can make for wheezing or trouble breathing. So that would be an indication to go and see your doctor as well. Just a few other other findings of leukemia that are hard to describe in words. So I wanted to show some pictures. This is a skin finding that we call petechiae, and petechiae are a specific type of skin rash that's caused by not having enough blood platelets. And these are just tiny little hemorrhages into the skin. And you know, lots of kids get these little red rashes from time to time. This one is different in that a normal rash, if you press on the rash, it gets white. And then after you let go, it you know, gradually gets back to being red. These little dots, you press on them and they stay red. So that would be the difference in distinguishing a petechial rash from a regular rash. Big lymph nodes is another potential sign of leukemia. Of course, kids can get swollen lymph nodes when they have an infection, but these are usually hard and firm and pretty apparent, like on this small child here. And then the final thing I'll show is pretty uncommon, but the specific type of leukemia that was reported in Dr. Tatton-Brown's paper can sometimes present with an infiltration of those cells into the gums, causing these puffy gums. So I've had some patients who have this finding and their doctor will send them to a dentist thinking it's just a gum problem. But if this occurs in your child, it might suggest a blood leukemia. Okay, so just to sort of wrap up my part of the talk, overall, we only found very subtle differences. By and large, the values we got were within normal range. There were just slight differences between the TBRS individuals and then the individuals without TBRS. Know your normal. I think that's important. Have a normal baseline so that you have something to compare it to. Should you have anything concerning arise that prompts your doctor to get another blood cell test done. Weight control might be helpful. Again, we think perhaps that increased neutrophil count we saw is a reflection of having increased basal inflammation. And one way to help reduce that would be weight control. Antibody levels could be of concern, so that may be something you should have your doctor think about, especially if there have been recurrent infections. And then really uncommon, and I don't think it's going to impact a large percentage of individuals TBRS, but I think the most important thing is recognizing the warning signs of a cancer so that you could go seek medical attention as soon as those become apparent. And with that, I'll invite Ayala back over to wrap things up. So I'll let you lead off this.
Thank you. That was really good. So, oops, no, too soon. We really thank you for listening to us, and we're really looking forward to all the questions. But just to summarize, of all the research here in Baylor, the blood focus, the weight, the brain, we really want to touch a few points. So we are studying the weight regulation and how weight gain is affected in TBRS. As Rachel also pointed out, it might also affect the blood by creating an expansion in the neutrophils. So we think for now that it's controlled not just by the connection of the brain and the fat tissue, but it also can be the process of other metabolic organs. We are exploring therapeutic avenues from our research, but we're not there yet. And we feel that it's going to take some more research from all of this collaborative research from all the groups that are working on TBRS, including us, to get there, but we're not there yet, unfortunately. A really important point that Rachel made in her talk, and also was really a key point from the study of the Mosaic individual we did, is that the mutations in the blood are really well tolerated, and not everybody, as Rachel said, will go to that avenue for a cancer development. And I think a key point that I want to say is that TBRS research is a marathon. It's not a sprint. It's long-term, it's surveillance, is your work with us. We can't do the work without you. And it's really a collaboration between us researchers, physicians, and other researchers or scientists, and you guys to go there, and it's ongoing. So anyone who wants to contribute or join our research, we're still recruiting. So just reach out to us. And just know that we're there for you. If you have any questions on the blood or cancers or doctor stuff, you go to the doctor. And if you have any questions on research, just email both of us. You have our emails, and we look forward to talking to you. We really want to acknowledge all the people who did the hard work in Peggy's group and Rachel's group. And those are the people noted here. I really want to thank Abe Elias, who is head of genetics in the Schroeder Children's Hospital in Montana. He's been a great help advising in genetics and other phenotypes or symptoms that we see in children with TBRS. We really want to give a big shout out and a huge, humongous thank you for Jill and Carrie and all of the TBRS community. It's been great working with you, and it's made our work so enjoyable, and we really have a wonderful time communicating with them and joining forces in this research. So thank you, and we look forward to your questions.
Thank you.